James Poulter. So I head up the molecular neuroscience research group. So I'm a research-based scientist. I'm not part of the diagnostic lab. I'm not part of the NHS or diagnostic testing network, or you won't see me in a clinic. I'm generally kind of hidden away behind the scenes. But I'm predominantly interested in identifying mutations that are underlying inherited disease. And obviously we've heard quite a lot over the last couple of days about the importance of this, not only for kind of carrier testing and getting a name or a diagnosis, but also for entering into clinical trials, all of which so far have required a mutation to be identified. But what happens to those individuals who don't get a genetic diagnosis? So, when you've been through the whole NHS testing system and essentially come back with nothing. And that's essentially where I come in. From a research side of things, we can take that genetic sequence and do more than what can be done in a simple kind of standard NHS test. So I thought what I'd do actually is, we're generally very good at saying, you should have a test and this is what your result is, but we don't generally tell you about your own genetics and what your genome looks like and how things work. So I thought I'd take this 20 minutes to do that, just so you get an insight into what we're actually looking for, the complexities behind it, and why it's not always quite so simple. So I'll briefly talk about genetics in your genome, about genetic variation in general across the genome, and then I kind of talk a little bit about genetic counselling and genetic testing for inherited disorders. So this is a talk that essentially applies to all genetic disorders, but I'll obviously focus on the kind of neurological and leukodystrophy sides of things. So your body is full of cells, and all of these cells are essentially very similar to each other, but they're all kind of very highly specialized. So you have brain cells, you have blood cells. They all do slightly different tasks, but they all, from a scientific point of view, look the same. And it's a little bit like the human population. We're all people, but we all have different roles, and that's essentially exactly the same in your body. The only difference is you have about 10 trillion cells in your body. Obviously, we don't have quite that so many people in the human race, but all of these cells, they all have a nucleus pretty much, they all have a mitochondria, they all have the same pretty much kind of subcompartments that all function in the same way. They're all just slightly specialized to do a particular task. Now, obviously, all of our DNA is in the nucleus. We've seen a very similar picture from Stefan of a carrier type. Now, they don't all line up nicely like that in your nucleus, but as you can see, there are chromosomes, okay, and there's 22 of these that we have 2 copies of each in general. And you can see we have chromosome 1 at the top left, chromosome 2, and there's two sticks to each chromosome, and essentially one of them comes from your mum and one of them comes from your dad. Okay, so we get a copy of each gene from each parent. Now in the bottom right, you can see one kind of on its own there, so that's the X chromosome. Okay, so it looks like the other ones when it's got 2 equal size chromatids, and obviously if it was a male, then there'd be a kind of a large chromatid and a slightly smaller one, which is the Y chromosome. So that's how we can tell genetically the difference between a male and a female. Now your DNA is in all of these chromosomes. We can essentially unravel it, and this is where all of your DNA lives, and each kind of gene lives in a particular place on a particular chromosome. It's a little bit like if you've ever done knitting. I haven't done it for a long time, I can assure you, but when you buy wool, it's all very nicely wrapped up, and you can never kind of quite get it back wrapped up that well ever again. And your genome is essentially exactly the same thing. It's also quite long. It's quite surprising. If you take one of your 10 trillion cells and stretched out all the DNA in there from one nucleus, it's about the same height as me. Okay, so from one tiny little cell that you can only see down a microscope, you stretch that DNA out, it's the same size. It's about 6 foot of DNA per cell. Now there's 10 trillion cells in us. Okay, so we've done the maths just because we can. So that's about 10 billion miles of DNA that's in our bodies. Now if you think going to the sun and back is about 90 million miles, so that's about 61 times to the sun and back. Okay, so that's how much DNA we have in us, in every single person. Now obviously it's really, really thin. So that's why it can do it. So you can stretch it out a long way, but it's really tiny, and you wouldn't actually see it if it was stretched out. But it's amazing. There's tons of it. And this DNA essentially encodes genes, so there's genes in there, and these genes tell our bodies how to make proteins. And the proteins are the functional things in our cells. Okay? So the proteins are the enzymes. They might be a kind of a channel on the surface. It might encode anything that our body needs to function. And each of these genes lives in a particular place on a particular chromosome. So we know what chromosome 1 looks like, we know what genes are on there, and it's the same pretty much for everybody, and the same for chromosome 2 and onwards. And you see, we can kind of open up these strands of DNA, and there'll be a gene here and a gene there, and each one of these genes is encoded by your DNA. Okay? So there's 4 bases, you've probably heard this, A's, C's, G's, and T's, and it's the order of these that essentially tells the body how to make each individual protein. And it's the combination, it's a little bit like Morse code, different combinations of letters encode different letters, which encode different proteins when you put them all together. So individual variants, so a single base change in a single gene, can affect how these proteins are made. It might make the protein incorrectly. Okay, so it might change it very subtly, and that might change how it functions, so it might not behave how it's supposed to behave. Now, sometimes these changes can have no effect whatsoever. We've actually got thousands of genetic variants in us. Most of them do absolutely nothing. They're just in non-coding regions, or they just have no effects. They don't impact the protein in a way that it damages it. Sometimes they're positive. We've all heard of evolution, and positive variants help us to survive better and survival of the fittest, and these positive kind of variants can help the human race survive. Also, there can be negative impacts, and this basically means we're stopping the proteins from working. So they have a particular job to do, and if that protein isn't doing that job, then that's going to have a knock-on effect on how the cell functions. Now, it can change, again, its behavior in different ways. It might change the shape of the protein. Okay, you might have heard of this lock and key mechanism for enzymes, and if an enzyme isn't the right shape anymore, it might not recognize the substrate that's supposed to connect to it. It might change its behavior. It might change where it is in the cell. Most proteins live in a particular place in the cell, and the variant might actually mean instead of it going to the nucleus, it goes to the cytoplasm, or instead of it being on the surface of the cell, it's now kind of buried deep in the cell, so its partners can't find it. Or sometimes it can even just degrade the protein altogether so you don't have any of it. And so it's these changes in the proteins that actually lead to the changes that we see in people. Okay, so it's not necessarily the genetic variant itself, it's the protein that's the functional unit of what's in our cells and the observations that we see. But sequencing proteins is not straightforward. There's lots of them, it's complicated, and actually sequencing DNA is much easier. So we sequence DNA, and then what we have to try and do is then predict how the variants we see in DNA impact on the protein. Okay, and this is why things start to get complicated. So, if we see a particular variant, what effect does that have? Would it cause the trait that we see in somebody if it was to, not function correctly? So, if you have a protein that controls eye color, the prediction would be if you alter that protein, then someone's eye color would change. And it's the same with leukodystrophies or any inherited disease. If you see a particular clinical feature, if you see a change in a protein, can we link that with what we see clinically as well? But actually this leads to then a lot of uncertainty. And herein lies the problem. And obviously, if you're doing a diagnostic test, especially in the kind of the NHS, we have to be let's say 99.9% sure that a result that's going back to a patient is the correct result, and we're not giving a false positive result, and therefore, enacting a change in the clinical treatment or the clinical care of an individual based on an incorrect genetic finding. The same with clinical trials. If we're trying to replace a gene that we think is faulty and it turns out it's not that gene that's faulty after all, then we can probably do more damage than help. So we've got to be very careful about how we define a variant and whether we say this is pathogenic, this is damaging, or whether it's not that damaging. And on the NHS, there's a whole system, a very complex system of how much evidence we need to be confident enough to relay a variant back to a patient as being pathogenic. And unfortunately, what happens is most variants then get classified in that yellow middle space, a variant of unknown significance, which basically means we're not sure what it does. And we don't have enough evidence to give that information back and for it to have an impact on clinical care. And so much of the research that we're doing is taking those variants and trying to gather enough information to say, is it having an effect or is it not having an effect? And can we then kind of relay that information back to the clinician and to the patient? So you've heard that the NHS now does whole genome sequencing. It's part of the routine kind of clinical care pathway that they're offering now. It's the first national health system in the world to offer this. And so this is a way of looking at the entire genome. So only 2% of our genome actually encodes for proteins. The rest of it used to be called junk DNA. Actually, it turns out it all does quite a lot of very important stuff. It's just we don't understand what it does on most, so we just call it junk DNA because it's better than just we have no idea. And so what we're doing is basically looking at these whole genomes of an individual and saying, where does it differ from the general population? Okay, we know what these genes are supposed to look like, and what do they look like in this individual. And so the sequencing is easy. In about 24 hours, we can sequence a whole genome. It's the interpretation of what we're seeing that's complex and challenging. And that's what's leading to the delays in getting test results back. I don't know if it's about 18 months now is the turnaround time for whole genome sequencing in the UK. And that's not because it's difficult to sequence DNA, it's because it's difficult to interpret what we're seeing in those genetic findings. And the problem is each person has around 13 to 14,000 variants in their protein coding DNA. Okay. All of these alter the protein in some way, or a protein. And so how do we identify which one of those, because it's most likely one, maybe two, that's actually causing an underlying condition? We have to basically filter out as much as we can, and we're not often left with just, oh, this is the variant. There's usually some kind of interpretation required. Now, we can make things a little bit easier for ourselves, so as part of the sequencing process and the screening process, we can just look in genes that we know are already associated with that particular phenotype, because that's where we're most likely to find a particular variant, but it's not always the case. And obviously, if it's in a gene that's never been identified before or associated with a disease before, then it won't even get looked at in the first place. But luckily, a genetic diagnosis is not required for genetic counselling. And obviously, genetic counselling is very important. It's in terms of predicting future pregnancies and kind of planning future care. And we can do a lot of this just based on normal inheritance patterns that we know exist. So you might have a dominant inheritance pattern, in which case you only need one copy of a mutation to cause disease, and you either get that from mum or you get that from dad. And because we each have 2 copies of a chromosome, if you see the red one, can I do it on here? The red one up there is the one that carries the mutation, and in dad, he's either going to pass on the one that has the mutation or the one that doesn't have the mutation. So there's a 50-50% chance that he passes that on to his child. Okay, so we can do that without even knowing what the genetic cause is. We can predict that risk. Whereas if you have a recessive disease, you need to inherit a copy of the mutation from mum and one from dad. Okay, and the chance of doing that means you have to have 2 carrier parents, and there's a 1 in 4 chance that we know then that both carrier parents pass that on to their children. So we know that for a recessive disease, there's a 1 in 4 chance if both parents are carriers that they will have an affected child. And so again, we can do all of this without any kind of knowledge of the underlying cause and genetic variant. Slightly different for X-linked disease, but again, we can still do the prediction testing based on known patterns of inheritance, of whether you need to have 1 copy of the mutation or 2 to result in a disease phenotype. And again, there's a slight difference in terms of the gender. Males only have 1 X chromosome, so if you inherit that a mutation on the X chromosome from mum, then that phenotype is going to be kind of come out in males with just 1 copy of the mutation, whereas in mum, they have a normal copy as well, so that means they don't necessarily get that disease. So we can do all of this kind of estimations and kind of family planning without actually knowing the genetic cause underlying the condition. So genetic counsellors, many of you will be familiar with these. They work directly with families. They're not clinically trained, but they work with the clinical teams to try and kind of make and support informed health decisions. And so they're able to help with the diagnosis, to suggest genetic tests. They help to manage and screen for particular genetic disorders So they're really kind of good stopgap in your kind of clinical support network. And so they'll use this information, such as family history, to try and predict whether you have a dominant disease or a recessive disease segregating in your family, whether it could be X-linked or it could be mitochondrial, and they'll take like a whole family history as much as possible to try and see if we can make the best predictions possible. And so you might see things like these. So we have very strict rules for how we draw family trees with males and females. This is a nicely drawn one, usually from a clinical geneticist. They're absolutely horrendously scribbled on a piece of paper, and you can't tell a square from a circle. But we can use these information to then say, okay, look, this is the pattern of inheritance in this family, and then predict, kind of using what we know about genetics, and we've known for a long time, what the risk is to future pregnancies. And again, you don't need to know the underlying genetic cause for this, but obviously if you do know the variant that's segregating in your family, then it does make things a little bit easier because we can do extended carrier testing. So these are the slides that we actually try and educate our medical degree students, so the people who are training to become doctors. They don't realise at that point in their training how important genetics is, and so most of them just keep asking why we're doing this, and it's actually, they don't realise how important genetics is until it's too late. And again, with X-linked conditions, we can look at, what happens if it's, the future child is going to be a boy, and what happens if it's going to be a girl, and what's the chances of someone being a carrier. So although we don't know the genetic cause, we can still make pretty good kind of educational guesses as to what the risks are for future pregnancies. But disease history and prognosis may be better understood if we can find the underlying genetic cause. And this is because we can then compare the prognosis with other families who have gene mutations in the same gene. Thank you. And so, and obviously with things like therapies, if we're having gene therapies, we need to know which gene is affected, which protein is affected in your genome, before we can even start thinking about correcting that. So but knowing the underlying genetic variant doesn't always alter the clinical management. It might give you a name, it might give you a feature or something to go away and look up on the internet, which is what a lot of families do. But obviously the more information we know about your underlying condition, then the better that is and the more kind of informed decisions we can make. So, in summary, that was a very brief introduction to genetics. So, your genome is long, it's about 3 billion bases, and long in terms of it can go to the sun and back 61 times. The interpretation of variation is complex, and this is a huge stumbling block in modern genetics. It's the same for every disorder, for every gene. Whole genomes are now being sequenced as part of routine clinical care, and this is included in the Inherited White Matter Disease Service that's just established in the UK. And a clinically relevant variant may not be identified or reported, and we need to make sure that you're prepared for that. And that the plan is, as Lydia said yesterday, is that if you do get a negative result back, then we can automatically enroll you into a research study like mine, which then can take that data and do a lot more kind of analyses that you don't get on a standard NHS genetic test. And obviously you can have genetic counselling to help with making informed decisions, and this can even happen even if a variant hasn't been identified. And knowing the genetic cause of disease may alter your clinical care, but it doesn't always, and that's, again, something that you need to be aware of. So thanks for listening. I hope that was useful, and happy to take any questions at the end or catch up over lunch as well. Thank you very much.